Assalamu alaikum dear students, I am Muhammad Rauf, lecturer Department of Zoology Cast. Welcome to the course Principle in Animal Life 2. This is lecture number 18. The topic is linked genes. Discussing agenda for the topic is linked genes, how linkage affects inheritance, genetic recombination and linkage, recombination of unlinked genes, and recombination of linked genes. Lecture outcome. After watching and listening this lecture, students will know about linked genes and how they violate Mendelian laws of genetics. Now, introduction to linked genes. Genes located near each other on the same chromosome tend to be inherited together in genetic crosses. Genes are located on chromosomes. The seat of a gene on a chromosome is called loci. So a chromosome may carry thousands of genes. Some of the genes will be located closer to each other. Other genes will be located wide apart from each other. So when two genes determining different traits are located closer to each other on a chromosome, then a crossing over event cannot separate them, and such genes will be inherited together. Suppose a gene controls skin color and another gene controls hair color. Now these two genes are located very close to each other on a chromosome, so a crossover event will not separate them and they will become part of the same gamete. Such genes are said to be genetically linked and are called linked genes. You should differentiate the term linked genes with sex-linked genes, because sex-linked genes refers to a single gene on a sex chromosome, whereas linked genes are the genes which are located closer to each other on a chromosome. So linked genes refers to two or more genes on the same chromosome that tend to be inherited together. Results of linked genes in breeding experiments deviate from Mendelian laws. In the second law, Mendel stated that the factors responsible for determining different traits are segregated independently from each other. He used the word factor for genes, and he said that a particular factor which controls seed shape will segregate independently of another factor which determines seed color. But here linked genes violate this law. This linked genes is an exception. They cannot segregate independently because they are located too close to each other to be separated by a crossover event. So linked genes or gene linkage is a violation of Mendelian genetics. Now, how linkage affects inheritance. Let's see how linkage between genes affects the inheritance of two different characters. T.H. Morgan chose two characters in Drosophila. one was body color and the other was wing size. Now the wild type flies have gray body color, so means the gray body color is a dominant trait, and they have normal sized wings. This is also a dominant character over a doubly mutant flies means they had mutations in the gene responsible for body color and the gene responsible for wing size, so they were doubly mutant, and they had black bodies and vestigial wings are considerably smaller than normal wings, so he took those two types of flies. One was homozygous dominant for body color and wing shape, the other was homozygous recessive for body color and wing shape. And neither gene was present on sex chromosomes. So there in that experiment there was no effect of the sex involved on the progeny. He wanted to assess only the inheritance pattern of linked genes, and those genes were present on somatic chromosomes. So in his investigation of these two genes, Morgan carried out the cross, which we will discuss on the next slide. The first was a P generation, parental generation cross to generate F1 dihybrid flies. And the second was a test cross. So he performed two types of crosses. The first cross was a parental generation cross where the gray bodied and normal size winged Drosophila were crossed with doubly mutant flies, and the F1 hybrids which he yielded were not crossed with each other, but they were crossed with the recessive parent. It was a test cross. Test cross is the cross between F1 hybrids and homozygous recessive parent. Now the resulting flies had a much higher proportion of the combination of traits seen in the P generation flies called parental phenotypes than would be expected if the two genes assorted independently. Means the F2 generation progeny, many individuals had the same genotype as that of the parents, means the grandparents, whereas very few individuals had other allele combinations. So a typical cross where we assume that the genes will assort independently gives a different F2 progeny as compared to when the genes are linked. So he concluded that the progeny achieved in F2 generation is different from the one which is expected for a cross between the two genes that assort independently. So Morgan thus concluded that body color and wing size are usually inherited together in specific parental combination because the genes for these characters are near each other on the same chromosome. Here you will have a better idea of his experiment. So in the parental generation, P generation, He took a wild type fly which was homozygous dominant for body color and wing size. Body color was gray, which is a dominant trait, and normal wing size means large wings. These two characters are dominant or over the homozygous recessive flies, which has black body color and vestigial wings, small wings. So he crossed these two flies. It was a dihybrid cross where the inheritance of two characters was under investigation, and he achieved F1 hybrids. Now it was a typical Mendelian result where the F1 hybrids showed the phenotype of the dominant parent. Now in second experiment, he did not cross the F1 hybrids with each other, but he crossed the F1 hybrids with homozygous recessive parent. Now a question arises that why he did not cross the F1 hybrids with one another, because he wanted to study the inheritance of these two alleles and the inheritance of these two alleles. If he had crossed them with one another, then it would have been difficult to assess the inheritance of the traits. Therefore, he crossed them with homozygous recessive parent, where here both the alleles are homozygous recessive and they are heterozygous recessive. So from that experiment, the inheritance of these two alleles to the next generation can be assessed as easily. So the results of the test cross were these are homozygous recessive, so only one type of sperm will be produced, and these are heterozygous dominant, so here different combinations are possible. This B can match with this one, then this B with this one, this B with this one, and this with this one. So all four types of combinations are possible here, and the expected ratio for F2 generation was 1 ratio 1 ratio 1 ratio 1. It happens only when the alleles are farther apart from each other, means if independent assortment occurs, then all the offsprings will have the same ratio. But if there is linkage between the alleles, if the alleles determining different traits are closely located to each other, then they will not be separated from each other. So in that case, there will be more individuals of the parental type, either this type or this type, and there will be very few individuals having a new allele combination. So what was the results he actually obtained? 965 individuals were of wild type, that is gray body and normal wings. This is a parental phenotype. 944 individuals were of black vestigial phenotype, which is also a parental phenotype, and these two were new allele combinations. You can see here one of the trait which is body color is dominant in that individual, but the wing size is reduced, vestigial. So this particular phenotype and this one are not found in the parents, but these two are the parental phenotypes. So he called them parental phenotypes which resembled the parents in P generation, and the new allele combination was known as recombinants. He expected that there will be no recombinants, but some individuals of this phenotype also were reported. Here is another illustration of the experiment. Morgan's experiment also resulted in production of both of traits not seen in P generation. They are talking about these two. These are new allele combinations or recombinants which are different from either parent. So these were non-parental phenotypes, suggesting that the body color and wing size alleles are not always linked genetically, and genetic recombination was observed. Means although majority of the individuals fall in the category of having parental phenotype, but very few individuals having a different phenotype were also reported. So it means that although the genes for determining the body color and wing size are located closer to each other, but still a certain degree of crossing over has taken place, which has resulted in the new allele combination. So genetic recombination is the production of offsprings with combination of traits that differ from those found in either P generation parents. This is genetic recombination, and the phenotype of those two are different from either parent. Genetic recombination and linkage. So meiosis and random fertilization generate genetic variations. These two are mechanisms for bringing variations in the offsprings, and this is because of independent assortment of chromosomes. If there is no independent assortment of chromosomes, then the same traits without any variation, without any change, will be transferred to the offsprings. So meiosis and random fertilization generate genetic variations among offsprings of sexually reproducing organisms due to independent assortment of chromosomes, and crossing over in meiosis 1 and the possibility of any sperm fertilizing any egg. So these are the various mechanisms which are related with sexual reproduction that results in genetic variability. Here we will examine the chromosomal basis of recombination of alleles in relation to the genetic findings of Mendel and Morgan. So chromosomal theory of inheritance was presented back in 1902 by Walter Sutton and Theodore Boveri. In that theory, they postulated that the behavior of chromosomes is parallel to the behavior of Mendelian factors. So here we are going to examine the chromosomal basis of recombination, that what happens at the chromosomal level and how the recombination of alleles takes place. Recombination of unlinked genes. Unlinked genes are those genes which are located far apart from each other, and there are chances of them being separated during a crossover event. So unlinked genes are assorted in a typical Mendelian way, and they show independent assortment. Now Mendel learned from crosses in which he followed two characters that our offsprings have combination of traits that do not match those of either parent. If you remember in face-to-face interaction in one of the classes, I said that Mendel reported certain crosses which he himself was unable to explain. Therefore, he didn't confuse the readers, and he didn't discuss them, because these unusual findings would have decreased the worth of his laws. So Mendel also presented or also reported such cases which violated even his own laws. So Mendel reported that some offsprings have combination of traits that do not match those of either parent. For example, consider a cross of a dihybrid pea plant with yellow round seeds, heterozygous for both seed color and seed shape. He performed his experiment with different traits, and in one of the experiments he crossed two plants, one having yellow round seeds which was a dominant character, with another plant which was having round wrinkled green seeds which was a recessive trait. So this cross acts as a test cross because the results will reveal the genotype of gametes made in the dihybrid plant. Now, in that experiment, the F1 hybrids were not crossed with each other, but they were crossed with the homozygous recessive parent, and it was termed as a test cross. Here, one half of the offsprings are expected to inherit a phenotype of the parental generation originally crossed to produce the F1 dihybrid. Means 50% of the individuals will resemble with either parent of P generation. So these matching offsprings are called parental phenotypes or parental types, but two non-parental phenotypes are also found among the offsprings. Because these offsprings have new combination of seed shape and color, they are called recombinant types, or recombinants for short. Recombinants mean they had new allele combination. When 50% of all offsprings are recombinants, as in this example, geneticists say that there is a 50% frequency of recombination, means some of the in some of the gametes, some of the cells that underwent meiosis, crossing over took place, and in other cells there was no crossing over. The predicted phenotypic ratio among the offsprings are similar to what Mendel actually found in his cross with the homozygous recessive parent and heterozygous dominant F1 hybrids. A 50% frequency of recombination in such test cross is observed for any two genes that are located on different chromosomes and thus not be linked. Means if the chromosomes are located far from each other, then the crossing over event can separate them and they may end up in different gametes, but because there were 50% parental phenotypes, so we cannot say that there was no linkage, absolutely no linkage, because if there was no linkage, then there would have been new allele combinations. So linkage to a certain point was there. That's why the parental phenotype was also reported. Recombination of linked genes, crossing over. Now let's explain the results of the Drosophila test cross. Recall that most of the offsprings from the test cross for body color and wing size had parental phenotype. 965 and 944, they had the parental phenotype. That suggested that the two genes were on the same chromosome, since the occurrence of parental types with a frequency greater than 50% indicates the genes are linked. So their frequency is greater than 50%. If you calculate, sum up the number of the F2 offsprings, it is equal to 2300, and about 1900 of them are parental types, and some of them have new allele combination. Why, if linkage exists, then why there is new allele combination? Why not all the offsprings shows the parental phenotype? It is because in some of the gametes crossing over do takes place, which separates the two alleles from each other. So how this recombination frequency is calculated, we will show you in the coming slide. Morgan proposed that some process must occasionally break the physical connection between specific alleles of genes on the same chromosome. Means to a certain extent the crossing over do takes place, which resulted in the new allele combination. Later experiments showed that this process, now called crossing over, accounts for the recombination of linked genes. In crossing over, which occurs while replicated homologous chromosomes are paired during prophase of meiosis 1. You already know that process in which the homologous chromosomes align with each other, gene to gene, end to end, through synaptonemal complex, and then they exchange segments with each other. In effect, when a single crossover occurs, end portion of two sister chromatids change places with each other. So this is the experiment which Morgan performed, and we analyze it on the level of chromosomes carry genes, so this is the homozygous dominant parent, two chromosomes are shown. This is the homozygous recessive parent, two chromosomes are shown, diploid individual. Now gametes will be formed, and the F1 hybrid will be heterozygous dominant. You can see one copy from the dominant parent, one copy from the recessive parent. Now in that individual, the process of gamete formation will take place, and during gametogenesis, crossing over, the chromosomes will first be duplicated, and then there will be a crossing over between the duplicated chromosomes. So you can see here these chromosomes have exchanged segments with each other. On the other hand, the homozygous recessive parent is having both the alleles of the same type, so here a crossing over will not make any difference. But here, because the alleles are different, so the crossing over will help them shift positions. Now four different combinations are produced here, and a sperm will be only of a single type, and then there is a simple cross. So these individuals are of the parental phenotype, and these are recombinants because crossing over occurs to a certain extent, and in majority of the gametes no crossing over occurs, therefore parental phenotypes are reported. So how recombination frequency is calculated? Write the number of recombinants or new allele combination or non-parental type 206 plus 185 makes 391, and divide that value by the total number of offsprings in F2 generation. You will sum up all the offsprings, and then you will divide the recombinants over that number, multiply by 100, so you will get 17%. So this 17% is calculated, and the recombination frequency is 17%. So remember that greater the recombination frequency, smaller will be the number of greater the value of recombination frequency, then there will be greater distance between the two genes, and crossing over will takes place. If the recombination frequency is lower, then it means the genes are located closer to each other, and the distance between them is minimum. So these values matter. This was the end of the topic. Thank you very much. Allah Hafiz.